So one of the things that I'm going to do during the course of this lecture is share a little bit with you about the inspiration that I've had from my mentors, my teachers, that have really inspired me along the way. And I'm going to tell you about my research journey. I'm going to tell you a little bit about the research that I've done in breathing and also where that sleep bit comes in. So these are the first two people that inspired me, and I know there are some people in the audience who they also inspired. This is my Professor Abe Guz. We did become Abe's babes, I'm sorry, there was lots of us. And Lewis Adams, and they were the people whom I originally worked for or with when I did my PhD. And the area of research that I was interested in while I was doing my PhD was breathing. And breathing's a really, really interesting thing to study because we all do it. So we all have an interest in, how is it controlled? Why do I take a deep breath? Why do I take a shallow breath? And words to do with breathing are very much in our culture. So it was really fascinating to start to learn a little bit more about breathing. Those of you that, I know there are singers in the audience, people that play musical instruments, how can you do that? How can you control your breathing to be able to do that? So the interesting thing about breathing, the thing that makes it unique, is that you can do it absolutely automatically. So if you just sit there attentively listening to a lecture, you will breathe. And yet, when you start thinking about your breathing, you're like, am I breathing slowly? Am I breathing shallowly? Shall I stop breathing? You can really control your breathing. And that's because your breathing has dual control. So you can do the volitional control of breathing because there's an area up here in the motor cortex that actually, we think, controls your ability to take a deep breath or your ability to regulate, change your breathing regulation. And it then sends those signals via the cortical spinal pathway to your respiratory muscles, to your diaphragm in your chest. And then there's also another area of breathing, which we think is somewhere down here in the lower brainstem at the bottom of your brain, and that's the automatic control of breathing. And that's the good thing about that, is that'll just go, it'll carry on working without you thinking about it. But breathing's the only system in the body that has this dual control, which makes it absolutely fascinating to study, because even in 2012, we don't understand how this cortical spinal tract and this bulbar spinal tract link together to control breathing. And you'd think we'd have worked it out by now, wouldn't you? But we still don't really understand how it happens. So when I went to the departments of medicine at Charing Cross, they were interested in trying to understand this control of breathing, because if you're a patient and you have difficulty in breathing, it's horrible. Those of you that have been struggled for breath with asthma and things, it's a really, really, really anxiety-provoking thing to happen. So understanding the control of breathing for those patients that have breathlessness or dyspnea is really important. Oh, that's interesting. It's now gone, oh, there you go. Obviously that button does that. So in the department, they were interested in investigating this area of the controlled breathing. And some of the people in the audience did some of these studies. And what they did was they used a magnetic resonance imaging to actually look for areas in this motor cortex that were actually controlling the volitional control of breathing. And these studies, which actually here published in 2003, are some of the first studies that identify this area of breathing, which actually is involved in breath holding and things. It's the first time that it's really been shown in humans where this breathing area is. Because you can't do these studies in animals. because as yet we haven't learned how to get animals to take deep breaths when we want to measure their breathing. So you can't do these experiments in animals. And yet the problem is when you start doing experiments in humans and you say, what I'd like to do is measure your breathing, what they do is they start thinking about their breathing and then they start playing around with it and you start doing deep breaths or light breaths. So it's actually very difficult to study these separate pathways in humans. And that's where the sleep comes in. Because by studying humans during sleep, we knock out this cortical spinal pathway. And now you become a brainstem preparation. So now you're a chemical preparation, and I can study your automatic control of breathing. Which is great, because when you go to sleep, we know that you're going to breathe all night. You do not go to sleep at night thinking, am I going to breathe all night? Somehow we just assume that when we go to sleep, our bodies are going to carry on breathing. Except tonight, where some of you might go to sleep and think, am I going to carry on breathing? But anyway, by and large, we carry on breathing, which is interesting actually from a sort of psychological point of view, because actually to asphyxiate somebody, to stop them breathing, is one of the most anxiety-provoking things that you can do to somebody. Having slightly high carbon dioxide levels in the blood is really, really anxiety-provoking. And so somehow we manage to suspend that anxiety and go to sleep. And that's good because it allows me to investigate your breathing. So why does that matter? Why am I interested in the control of breathing? Most of you go to sleep and you breathe. This is data from a patient. So these tracers at the top here are the sleep tracers. So this is the electroencephalogram, which is the brain activity, the top two. The next two down are the right eye and left eye movements, the ocular activity. This is your postural chin muscle activity. And these ones down here are the breathing, and these ones down here are the blood gases, the carbon dioxide and the oxygen. So this patient is asleep, and they're breathing nice and rhythmically. And then at this point here, something happens. They wake up from sleep. There's probably a sound. Maybe their mobile phone goes off, and you get a sort of increase in the breathing, and you're, oh, I've woken up. And all my signals start moving around. And then look what happens. Because they've woken up, and now they're beginning to oscillate in and out of sleep, you get this periodic breathing pattern. And those of you that do medicine in the audience will know that this periodic breathing pattern is called Cheyne-Stokes breathing, and that these periods here, where the person's not breathing, are called central sleep apneas. So it's central because it's a respiratory control problem in the brain. It's sleep because it occurs during the sleep-wake transition. And it's an apnea because apnea means cessation of breathing. So central sleep apnea is a problem with the respiratory control system when you go from sleep to wake. And so when I joined the team at Charing Cross, I'd seen these breathing patterns occurring in patients who'd got cardiac disease, patients who were having problems with their neural control of breathing. And I was interested in trying to understand why this breathing pattern occurred. But the question that you've then got to ask yourself is, how are you going to understand it? Because we can't actually do neural imaging. You know those MRI pictures I showed you? We can't do that during sleep because the MRI imager is noisy, and at the moment we haven't learned how to get people to go to sleep for long periods in MRI scanners. So we have to find another way of investigating this control of breathing to try and understand why these patients are breathing the way they are. And then, of course, if we can understand it, hopefully we'll be able to help them. So we're going to back up for a minute now, and I'm going to tell you a little bit about sleep so that you understand what we're going to be looking at in the patients. So these are the brain activity again, and the top line here is brain activity that I recorded at the scalp, and you can see that it's very low in amplitude and fast in frequency. So as you're all sitting there, attentive, listening to the lecture, you've got the fast frequency activity going on in your brain. And then you'll see that down here, the brain activity gets slower, and you get these larger amplitude waves. And this, rather unimaginatively, is called slow wave sleep. And it's the deepest stage of sleep, and that's the one that makes you feel better when you wake up in the morning. But going from wide awake to deeper sleep is a continuum. And in between times, you get these different stages of sleep that we call 1, 2, 3, and then there's 4 down here. And this stage up here, which is the transitional stage from being wide awake to deeper sleep, you can sometimes see people going through this stage, maybe on the back row when you're giving a lecture, or another time you can see it is when you're on the tube. So when you go home, those of you that are going on the tube, don't do it if you're on a bike, but you'll be able to see somebody, and what you do is you watch them, and what happens as you go to sleep is, first of all, your postural muscle activity starts to relax, so you'll see them do the head thing. And then what will happen is that the ocular muscles start to relax as well, and you get the slow rolling eye movement thing going on. But in those light stages of sleep, you're actually responsive to sensory input. So should you be a student and you're sitting on the tube, or even a lecturer, I would dare say, and you get to South Kensington and you're doing this kind of thing, and it goes South Kensington, you will look up, you will begin to look up. When you get down into these deep stages of sleep, you don't have sensory input. You become my brainstem preparation. And at that point, all bets are off. You're on the circle line, going round and round, dribbling. It's not good, that stage. Now, those of you that have actually jumped ahead in the talk will see that there's this type of sleep down here, which has got the title REM sleep next to it, which is rapid eye movement sleep. And that's the stage of sleep in which most of you will know you're dreaming. So the interesting thing about this stage of sleep for us is that you'll see that the brain activity is quite fast. And in fact, we call it an active stage of sleep. It's a really strange state. We don't really quite understand why we have it. But for respiratory physicians, it's important because in REM sleep, you have a functional paralysis of your muscles. You're not actually paralyzed, but your brain actually does stop you moving around during REM sleep, presumably. And so if we have patients who are having difficulty in breathing while they're awake, when they go into REM sleep, they have even more difficulty in breathing because they have paralysis, not of all the respiratory muscles, but of some of them. So REM sleep is a particularly difficult time for patients with respiratory disease. So now you're experts on breathing and on sleep. What we would then do is we would talk about how breathing is actually controlled so that you can understand what we're trying to study. So in these areas of the brain, you've got the cortical control and the brainstem control. And those two areas of the brain send neural muscles to the respiratory muscles in your chest, which causes your lungs to inflate. You breathe, which brings in oxygen into the body and gets rid of carbon dioxide. And so in your blood, you have a regulated level of oxygen and carbon dioxide. And those signals then go to the respiratory center, and the whole thing carries on. And you have this system that is very, very finely controlled. So why does it go wrong? Why do we think that this system has problems regulating itself at the transition between sleep and wakefulness? And what we think is happening is that there's a problem over here with the control of the blood gases during sleep. So when I was doing my PhD, I thought, okay, well, what we'll do is we'll study patients who've got difficulty in controlling their blood gases. And that's exactly what we did. So we collected a group of patients who had unilateral focal lesions in the rostrolateral surface of the medulla, and we looked at whether they've got problems with their chemosensitivity, and we measured their breathing during wakefulness, sleep, and exercise. And so many people have said to me, if I come to your lecture, will you understand it? And I said, yeah, I'll understand it, you'll understand it. So just so you all understand this lecture, I want to make sure that you're going to explain this slide. So these patients have difficulty in regulating their chemicals, their carbon dioxide and oxygen in their blood. And what's happened to these patients is that they've had a bleed in the basilar artery, which is down here. So this is your cortex over here in the brain. This is a slice through the brain. This is the medulla. And these patients have had a bleed in the basilar artery, which blocks off this posterior inferior cerebellar artery. I know there are some anatomists and some neuroanatomists in the audience, so just accept this bit for me. And what happens is that this ventrolateral surface of the medulla then has difficulty in getting enough blood to keep the brain cells alive. So if you do these slices through the brainstem, you can see that there are these grey areas here which are on the surface of the medulla, and these are the areas that we think control your chemical control of breathing while you're asleep. Well, they do it while you're awake, but while you're awake, if they don't work, you've got the cortex as a backup. But if you go to sleep, you lose the cortex, you become a brainstem preparation, and if this doesn't work, then there's going to be a problem. So we got together some of these patients. Fortunately, this disease is quite rare, so it took us a while to collect them. And this was the breathing pattern that we measured in these patients. So what you've got here are dips in oxygen saturation, and you're now experts, so you know that this person's breathing, not breathing, breathing, not breathing. So it seems like what we thought was happening was true. But those of you that know a little bit more will know that this measurement here, the esophageal pressure, is measuring the pressure in the chest. And what you can see is that during these periods of no breathing, some of the time they're not breathing because they're not making any effort to breathe because they've got that central respiratory control problem. But some of the time they are trying to breathe, they're making efforts to breathe, but there's no airflow. So how can that be? How can you make efforts to breathe and not get any airflow? So this is the audience participation time, right? You block your nose and mouth off and you try and breathe. I can see some of you are not doing it. You're not as good as the students. They all get involved. Now, if you do that, what happens is that you move your diaphragm up and down. Now, a few people are trying it. I can see that you move your diaphragm in and out, and you do what's called paradoxical breathing or fictitious breathing. It's a bit like going to the toilet. It's a bit like trying to breathe. But what happens is that if the muscles up in the airway block off, then you do make respiratory efforts to breathe, but you don't get any air in. And we call that mixed sleep apnea. We're not very good at thinking up imaginative names, I'm afraid. It's mixed because there's a central control problem and there's also a mechanical problem. It's sleep because it happens at the sleep-wake transition, and it's apnea because it's the cessation of breathing. So if you go back to your diagram, what happened with these patients is, yes, they had problems controlling their blood gases, but they also had problems controlling their respiratory muscles, and we did not expect that. That was not something that we expected. So it was a surprising finding. And one of the things that Abe Guz and Lewis Adams always taught me was expect the unexpected and be willing to interpret your data in the light of new findings. Really be open to all things educational. So this was a surprising finding for us. So what we demonstrated in these patients is that there was a threshold over which their breathing had to go in order so that they had stable breathing, their blood gases had to go to have stable breathing, but there was also a mechanical problem. And that threshold was termed, or is termed, the apneic threshold. It's the threshold over which your blood gases have to go in order to maintain breathing. But these particular patients also had problems with their respiratory muscles, and I'll come back to that in a minute. Because now we're going to go to America, because we've finished our PhD and we have to go to the next phase. And in fact, if you see, there's a name here that says Scatterud and Dempsey, 1983. And these were the guys that explained the apneic threshold. And these were the guys, in fact, that you will see in the next slide. Because off I went to America, finished my PhD, not quite Dr. Zeus, but Dr. Morrell. And as Sue said, I went to America, and I got what they called in the Department of Medicine your BTA. You need your PhD, Mary, and then you need your BTA, which is been to America. I told you we're not good on names. And I did go to a swing state. My job's been made easy, hasn't it, over the last couple of days? You now know you all know where Bruce Springsteen was, and Barack Obama, and Wisconsin is indeed a swing state. It's a great place. And this is the guy that I worked for. This is Professor Dempsey, who first described that apneic threshold. And this is actually the first time I met him. And the inspiration that I got from Professor Dempsey is for 2.5 years he used to go, "Speak up!" And what I got from that was that you have to be willing to communicate your science. You've got to defend your ideas. And it was a great training for me. And what I did while I was there was I studied the respiratory muscles. I went away from the chemical control of breathing and the brain, and I started looking at the mechanics of breathing. And so we started studying a type of disease which is called obstructive sleep apnea. So we've had central sleep apnea, we've had mixed apnea, now we've got obstructive apnea. And obstructive sleep apnea is much more common than either central or mixed apnea. It happens to about 4% of adult males and about 2% of adult females, and that makes it the third most common respiratory disorder after asthma and COPD. But because it happens at night, probably, less people know about it. And what it is that this area here at the back of the throat is a muscular tube. So in the front of the tube, or at the front areas, is your tongue, and then coming round the back are the pharyngeal constrictor muscles. And so if I take a slice through this area here at the back of the throat, you end up with just a straightforward muscular tube. And so what happens is that as you expand your rib cage to get air into the lungs, you generate negative pressure at the back of the throat here, and that negative pressure is pulling air into the lungs, but it's also sucking this part of the airway closed. So you generate the pressure to breathe in, and because the airway at the back of the throat is floppy and relaxed, because you all now know that your muscles relax when you go to sleep, the airway goes and gets sucked closed. So obstructive sleep apnea is the closure of the airway during sleep. So there's nothing wrong with the brain. The brain knows that you should breathe. It's controlling your breathing. But you can't get the air in because it's effectively blocked. There's a collapse of the airway. So you get negative intraluminal pressure sucking the airway closed, and you also get positive extraluminal pressure, the adipose tissue that some of us have around our neck, and the gravitational forces as you lay in going to sleep and your tongue falling back that causes your airway to collapse. So that's what happens in obstructive sleep apnea. And what we were interested in while we were in Madison is what phase of the respiratory cycle does this happen? Is it all to do with the airway being sucked closed, or is some of it to do with the airway being pushed closed? And how can we stop it happening? Because at the moment, there aren't any drugs that are available that keep this airway open. So we don't have any drugs that we can treat this disease with. So we thought what we'd do while we're in Madison is actually try and measure this airway closure. So we did that by doing this experiment. So what we did is we took a pediatric fiber optic bronchoscope and we put it down into the back of the throat in this area here at the back of the soft palate, which is the narrowest area, and that's called the retropalatal airspace behind the soft palate. And we then collected these video images with these respiratory movements from these two bands, and we measured the breathing. Now, this sounds really easy now, doesn't it? You could probably do it on your iPhone. But at the time, this was really cutting-edge technology, because acquiring the video images at the same time as acquiring the breathing signals, it took us months to work out how to acquire these signals onto the computer. But you know what? Now, you could just do it really simply. Anyway, I'll show you some old images that we collected at the time. These are images from a healthy person, and if I just click on it, it should run the video. There's no sound on the video. Oh, I've done that other thing. And what you can see is this is the soft palate that hangs down the back of the throat here. This is the epiglottis. And you can see that even though this patient is asleep and the airway is relaxed, they're still managing to get the air in because there's this patent airway lumen here. So the air is still going in and out of the airway because the airway is nice and big. So when you go to sleep tonight, even though your airway will relax and get a little bit smaller, you'll still be able to get the air in and out of your airway because you've got a relatively large airway to start with. But when we took patients who'd got obstructive sleep apnea and put them in the lab, this is what we found. So the first thing that you'll notice there is that the airway is moving around quite a lot. Actually, the first thing that you might notice is that there's quite a nice big yellow line down the side of it here, and that's that catheter that goes down into the chest and measures pressure. But you can see that the airway is moving, and what's happening is that that's just about to go. That's expiration, that's inspiration, that's expiration, that's inspiration. So each time the patient breathes in, the airway is getting sucked closed. But what I want you to notice is that during expiration, the airway is also moving. And in theory, that shouldn't be happening. That's expiration, that's inspiration, it's stopped now. But what we noticed was that not only was the airway getting sucked closed, but it was also closing during expiration. And in theory, that shouldn't have been happening because all the books, everybody said, the airway collapses during inspiration, not during expiration. But what we found was positive airway pressure pushing the airway closed during expiration. Which is interesting, but who cares really, whether it's inspiration or expiration. The reason that it's important is because the way that we treat these patients is in order to help them breathe at night, we actually blow air into the airway. So this is the treatment for obstructive sleep apnea. It's the best we've got. It's not great, but it's the best we've got, and it works. And what the patients have to do is they have to sleep with a mask on their face. So this is an adult and a babe as well. And then the mask is connected to this machine here, which blows air into the airway during inspiration and keeps it open. During expiration, what the manufacturers of these machines were tending to do is reduce the air, because if you try and breathe out against air coming in, it's uncomfortable. So they thought, well, we'll lower the pressure as they breathe in, and then ramp it up as they, lower it as they breathe out, but ramp it up as they breathe in. And so what we found was that actually you can't do that. You have to keep the pressure fairly high when you breathe in and when you breathe out. So the work that we did in Madison was actually incorporated into some of the algorithms that the companies who make these machines use in order to make sure that the patients have appropriate levels of air pressure when they breathe in and out. And although those video images look quite crude, they were the first time that that had ever been done. Okay, so it's now time to come back to the UK. Done our BTA, back to the UK. And as Sue said, I was very privileged because while I was in America, I was on a Wellcome Trust fellowship. And the thing about that fellowship is that it had both money in the US and then a year's worth of money back in the UK. And the guys that were in the lab before me that trained me, one of them went to Harvard and stayed there, the other one went to Canada and stayed there. And I'm pretty sure if I hadn't have had this fellowship, I wouldn't have come back either. So, hats off to the Wellcome Trust for setting up a scheme where it allowed people to go to the States and also to come back. And one of the reasons why I came back was because when I looked at this map of London, there was almost no centres in London doing the kind of research that I thought that should be done for patients who had obstructive sleep apnea. Remember, it's the third most common respiratory disease, and there's no labs, or very few labs in London, 5.5 million people, I think it is, who are doing research in that area. So I thought, I'll come back to London, I'll set up my research group, and I'll make a difference, and that's why I came back. And I looked around for people to talk to, and one of my mentors at the time was Professor Stradlin, deep in conversation here with one of my colleagues from St. Thomas's. And one of the things that Professor Stradlin said to me was, you've got to stay focused. You've got to decide what it is that you're going to do research on. You've done a bit of that chemical control of breathing, you've done a bit of that automatic control of breathing, you've done some muscles, decide what it is that you want to do. So I looked around at the time at what the big problems were. And at the time, the Wellcome Trust, and I think still absolutely are interested in, is this problem of the aging population. We're all going to get there, guys. And so, obviously, these are plots from German population. Very few. I've drawn the line at 85, because over 85-year-olds are the ones that use a lot of the healthcare resources. And if you look at over 85s in 1956 in Germany, there's hardly any. Getting to be a few more now, but look at this big bulge here. I also worked out that I'm probably in this big bulge as well. Seems like a good thing to do some research on. But also, the other reason why I was interested in it is because while I was in Madison, it was an amazing center for sleep research, and they had an epidemiology study going on at the time, and it was being done by this author here, Terry Young. And what she was doing is she was sequentially following cohorts of people in Wisconsin and looking at how they breathed during sleep every 4 years. And she published a study in 1993 with 600 people. And these people were state workers in Wisconsin between the ages of 30 and 60. And that's where the number of 4% of men and 2% of women having sleep apnea comes from. It may actually be higher now because sleep apnea is exacerbated by obesity, and of course we're in the middle of an obesity epidemic. So the prevalence of sleep apnea is probably more than 2 to 4% now. But Terry then went on to set up a couple of cohort studies with much larger numbers, looking specifically at older people. So you'll see these cohorts are 60 to 99, and one of them, this one's got an average years of 70. And look at this number. 20% of older people have difficulty in breathing at night. That's a lot of people. And in some cases, the numbers go up to a third. So definitely a problem that we should look at. So what we did is we thought, right, we'll see why it happens. So we measured their breathing during sleep. And this is the breathing in a young person. This is the breathing in an older person. And these are both healthy people now. This is awake, this is asleep. And what you can see is that this young, healthy person, they're breathing in and they're breathing out, and they're breathing in and out, and there's a nice straight line here, and they're managing to breathe fine. And so is the older person when they're awake. But when these people go to sleep, what happens here is they start to breathe in. This is airflow on this axis here, and this is pressure. So as they start to breathe in, they get airflow, and then at this point here, they're trying to generate respiratory pressure along this axis, but there's no airflow. And you now know that if you generate respiratory pressure and don't get an airflow, the only reason that that happens is because your airway's blocked. So what's interesting is that at the peak of the inspiration here in this young person, they're having difficulty getting the air in, but it's maybe not too much and it's not too bad. But look at this old person. They're hardly getting very much air in at all before the airway starts to close off. And what we found is that almost all old people have some difficulty in breathing asleep. It might not completely block off, but the airway is difficult to keep open. And we think that's because when you get older, things begin to sag and move around. And I'm trying not to look at anyone particular in the audience now. Just notice I've caught somebody's eye. How bad is that? And so what happens is that you get changes in the upper airway. And in particular, what happens is that there's a change in the position of the hyoid bone, which is the top of the larynx. And what happens is that the hyoid bone moves down and forwards, and that lengthens the esophagus and the retropalatal airspace here, and it actually makes it, we think, more susceptible to airway collapse. So the problem as you get older is your airway becomes more susceptible to collapse. And we thought that was really interesting, and we thought it was important, and we thought we should now start, we looked at healthy people, we should now start looking at whether that happened in older people, and if it did, should we be targeting them with treatment? So we were all set to go. Except life doesn't go that way, does it? And every now and again, things aren't what you think they are. And things don't always go the way you plan. And at this point here, we lost our funding. So we were going along brilliantly, and then other priorities, funding priorities, took over for the people who we were applying to, and we lost the funding. And when something like that happens, what you need are some wise words. And so this is one of the wisest women that I know. And what she said to me when this happened, this is such a girl comment. She said, "Mary, you've got to be comfortable with what you see in the mirror." And actually what I took that to mean is you're not ugly. You've really got to remain true to what it is you want to do, because in the end, there's only you that you're going to answer to. The other way of putting it is the more boy comment, which is the Winston Churchill comment, I think, isn't it? Which is never, never, never give up. So we decided to carry on going, and I did think that looking at breathing in older people was really important, but we decided to look at it from a different angle. So what happens in patients with sleep apnea is they fall asleep, the airways, the muscles relax, the airway closes. When the airway closes, you can't get the air in and out, so your oxygen levels fall, you get hypoxia, and your carbon dioxide levels go up, and you make efforts to breathe, and that wakes you up. So the patients don't turn up in clinic saying, "I've got difficulty in breathing, doctor." They turn up in clinic because these arousals make them sleepy, or because they're choking or their partner's seeing them choke at night. So we decided to tackle the problem, not from the mechanistic why it's happening problem, but what the consequences of this apnea were. And we decided particularly to focus on this low oxygen because at that time, some work had been published by our colleagues in North America, which said that if you subject animals to intermittent hypoxia, what will happen is that this area of the brain, which is the hippocampus, will start to get cell death, apoptosis. So in this area of the brain, the C1 area of the hippocampus, which is an area that's particularly susceptible to low oxygen, they found cell death in animals that had low oxygen. Which is kind of interesting, but we're not animals, or we are, but we've not been subjected to the same levels of intermittent hypoxia that these animals were. So we decided to try and study that in humans. And what we did was we put some people with sleep apnea into an MRI scanner, and we looked at their brain by this new technique that had then just come out called voxel-based morphometry. And remember, we haven't got much study funded at this point, so we decided we'd just study a small number of people. Now, this might not look like a very interesting slide to you, but it slices through the brain. So they're slicing down and across and then across the top. And you can see, I think, even with the lights being up, that there is an area here where these cross marks are, and that is the left hippocampus. And those areas there, that marking in red there, means that there's an area of cell loss. The neurons have died in that area, potentially. So this was very interesting for us, because what it means is that in our sleep apneic subjects, we've been able to show that there is some neural cell loss. However, before we book our slot on Radio 4 and decide that we're going to tell the whole world that if they've got sleep apnea, they've got cell death, we thought we'd better verify that finding and actually study it in more detail. So we collaborated with some of our colleagues in North America and in Australia who were doing the studies, similar studies at the same time, and we studied more people. So this time we studied 60 people with sleep apnea and compared their brains to 60 control people. And what you'll see here is that instead of the blobs being red, they've now turned yellow, which means that the neural loss is not quite as intense. There's not quite as much neural loss, but you'll see that it's now more diffuse. The blobs are bigger. And in particular, there's a large blob down here in the cerebellum. And that's interesting. It doesn't occur in this one over here, because at that time we couldn't actually test the area of the cerebellum. The imaging techniques weren't very good enough. But now what we've found is that this is the most recent data that we've got, 2010. There are large areas, or we think there are potentially areas of cell death in the cerebellum, and also in areas around the hippocampus, but not quite in the hippocampus. So that's where we are at the moment. So now what we decided to do is look for the functional consequence. So I'm not going to go into this slide in too much detail, but the idea of this slide was that we'd look at different sorts of memory and we'd try and equate it to those areas of cell death. So the idea is that if you've got a problem in the cerebellum, maybe some of these sorts of memory would be impaired. And what we found was that there was one particular sort of memory that was impaired. And this is verbal episodic memory. So episodic memory is specific things that you're told. So what's your first day at school, and what is your, when you got married. Semantic memory compared to it is things like, what's the capital of France? You know it, but you don't know when you first learnt it. So verbal episodic memory is the thing that sleep apneic subjects seem to be less good at. And that's interesting because to a certain extent, episodic memory is controlled by the hippocampus. It's also interesting because what's the first thing that any doctor does when they sit you down in clinic is they start giving you verbal information. So our poor sleep apneic patients have very much difficulty encoding that verbal information. And in fact, what we've done with this finding is that we have thought about other ways that we can deliver the information to our patients with videos or maybe with information they can take away. So the story so far is that obstructive sleep apneic subjects appear to have brain lesions that are in the hippocampus and in the cerebellum, and there do appear to be functional consequences to that neural loss. And that's where we are so far. Except that we started out talking about old guys, didn't we? And what happened to them? Well, what we think might be happening, and this is speculation, is that if you are older, and so therefore you've got cell loss anyway because you're getting older, the actual cell loss that you're getting may be exacerbated by the sleep apnea to accelerate cognitive decline. So if you're older, and then also if you're younger in children, you've got a particular vulnerability to that low oxygen, which may mean that the sleep apnea is more detrimental. So we thought what we would do is try and investigate that. So we set up this study. So this is a study which has been ongoing now for a couple of years with collaborators in Oxford, Edinburgh, the MRC, and also in York. And this is a study of positive airway pressure. You now know that's the treatment for sleep apnea in older people. And it's a randomized controlled study. So some of the patients get their treatment, and some of them get randomized to no treatment. And this is the best way to determine what a treatment impact is in older people. So if some people are not being treated, then maybe their cognitive decline and their sleepiness and the symptoms of sleep apnea are going to get worse. And if they are being treated, then they will actually not get worse. And this is an ongoing study at the moment. And one of the things that I am most proud of in this study is this map. So remember I said that when I came back from North America, there were very few labs working in this area, with the exceptions of places like Oxford and also Edinburgh. And now look at this map. This is a study funded by the NIHR, which is the research arm of the NHS. And what they did is they funded us to allow us to set up this network of centres, all of which collaborate together to contribute to the study. And although not all the centres, like Blackpool and Swindon, are not leading the study, they are contributing to it, and the patients in those centres are benefiting from being in the study. So, where does that leave us now? What's next? Well, we have to answer that question of yes, no, maybe. All our patients are recruited. We recruited over 300 patients, and it'll be about a year before we get the answer. So you can come back in a year if you like, and we do it all again. We've also got a couple of other new ideas in the pan. The first one is that the development of magnetic resonance imaging is going on all the time. And with some of my collaborators that are in the audience at the moment and at the Institute of Psychiatry, we can now not just measure those blobs of cell death, but we can look at the tracks of the cells as well to see how they're linking together. So we're going to do some of this imaging to actually look in more detail about how and why and which areas of the brain are getting influenced by this low oxygen level. And this is really exciting work. This is a great article if you're interested in that, which has recently come out in Science. We also are really interested in how to develop the technology that we're using. And I know some of you in the audience have done sleep studies for me. You are those healthy controls that I've been showing you. And when you do a sleep study, you get all this equipment on you. I measure your EEG up here with these electrodes. I measure your breathing. I measure your heart rate. And you know what? You don't love it, do you? You can't really sleep very well. It's awkward and it's unpleasant. So one of the great things about working at Imperial is that there are some really smart people here. And what we're doing now is we're trying to develop the technology to actually make sleep studies simpler. So we're going to take all these sensors here and we're going to put them into these little moles that go in your ear, like a hearing aid. And we're going to try and develop a sleep system. We've actually got quite a long way down the road where we can measure both your sleep and breathing by putting this gizmo in the ear. I'm really excited about that one. I think that one will be great. And what it will do is it will increase the access to sleep medicine for patients who maybe don't all live very near an specialist centre of sleep research. So will we succeed? This is the fun bit everybody's really waiting for. Yep, we'll succeed. These are the research people that have worked with me over the years, and I am absolutely indebted to them for doing this brilliant science that we've been talking about today. I'd also like to thank my clinical colleagues at Royal Brompton, some of whom are here today, because it's the synergy between the research and the clinical that really drives what we do in order to try and make a difference for our patients. There are still some people in the audience from Charing Cross, which is great. And these are the people that inspired me while I was at Charing Cross. And I'd really like to thank them for setting me up on this journey. I'd like to thank my colleagues at Imperial College, my teaching colleagues, and my administrative and all the people behind the scenes that actually mean that we can do the work. They administer the grants, they make a huge difference to us. They're also really difficult to get photographs off. So I'm sorry I had to nick some of these off the website. And you can't do it without those funding agencies. I spoke about the NIHR who are funding us at the moment. They've been great. The Wellcome Trust, British Heart, and the British Lung Foundation, who actually have got a drive on sleep apnea at the moment. We're almost at the end, guys. The last one is the friends and family one. These are the people without whom there would be no journey. And I am absolutely privileged to have you as my friends and my family. I love it. You join in every single mad plan I have, whether it be climbing mountains, whether it be running fundraisers, doing art projects, getting involved with the Olympics, or just generally something quite odd. You go for it, and I really, really thank you. And lastly, this really, really is the last one. I thank the patients and the volunteers that have allowed me to do my research. You make a huge difference in my life because I get to do a great job, but you also make a difference, hopefully, for the future if we can develop some of these treatments that I've been talking about and some of these approaches. Thank you.